friends, Unit 6 Reproduction. Chapter 1 Sexual Reproduction in Flowering Plants. Chapter 2 Human Reproduction. Chapter 3 Reproductive Health. Biology is essence is the story of life on earth. While individual organisms die without fail, species continue to live through millions of years unless threatened, threatened by natural or anthropogenic extinction. Reproduction becomes a vital process without which species cannot survive for long. Each individual leaves its progeny by asexual and sexual means. Sexual mode of reproduction enables creation of new variants so that survival advantage is enhanced. This unit explains the details of reproductive processes in flowering plants and humans as easy to relate representative examples relate perspective on human reproductive health and how reproductive health can be avoided is also presented to complete our understanding of biology of reproduction. Born in November 1904 in Jaipur, Rajasthan, Panchanan Maheshwari Panchanan Maheshwari rose to become one of the most distinguished botanists not only of India but of the entire world. He moved all Allahabad for higher education, where he obtained his BSc during his college days. He was inspired by Dr. Dudgaon, an American missionaries teacher, to develop interest in botany and especially morphology. His teacher once expressed that if his students will progress ahead of him, it will give him a great satisfaction. These words encouraged Panchanan to inquire what he could do for his teacher. In return, he worked on embryological aspects and popularized the use of embryological characters in taxonomy. He established the Department of Botany, University of Delhi, as an important center of research in embryology and tissue culture. He also emphasized the need of initiation of work on artificial culture of immature embryos. These days, tissue culture has become a landmark in science. He worked on test tube fertilization and intra-ovarian pollination, won worldwide acclaim. He was honored with a fellowship of Royal Society of London (FRS), Indian National Science Academy, and several other institutions of excellence. He encouraged general education and made a significant contribution to school education by his leadership in bringing out the very first textbook of biology for higher secondary schools, published by NCERT in 1964. Sexual reproduction in flowering plants. Are we not lucky that plants reproduce sexually? The merits of flower that we enjoy gazing at, and the scents and the perfumes that we soon have over, and the rich colors that attract us are all there as an aid on to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. I look at the diversity of structures of the inflorescences. Flowers and floral parts shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction. In flowering plants, angiosperms, flowers are fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbols of for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least five flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of five more flowers that are used in social and cultural celebrations in your family. Have you heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. In earlier classes, you have read the various parts of a flower. Figure 1.1 will help you recall the parts of the flower, typical flower. Can you name the two parts of in a flower in which the two most important units of sexual reproduction develop? Pre-fertilization structures and events. Most before the actual flower is seen on plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescence formed which bear the floral bud, and then the flowers. In the flower, the male and female reproductive structures, the androecium and the gynoecium differentiate and develop. You would recollect that the androecium consists of a whole of stamens representing the male reproductive organ, and the gynoecium represents the female reproductive organ. Stamen, microsporangium, and pollen grain. Figure 1.2a shows the two parts of the typical stamen, the long and cylinder stalk called the filament, and the terminal, generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number of length of the stamen are variable in flowers of different species. If you were to collect a stamen, each from the 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making the neat diagram would elucidate the range in shape and attachment of anthers in different flowers. That a typical angiosperm anther is bilobed with each lobe having two theca, that is they are diathecous. Figure 1.2b. Often a longitudinal groove runs lengthwise separating the theca. Let us lengthwise separating on the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. Figure 1.3a. The bilobed nature of the anther is very distinct in the transverse section of the anther. The anther is the four-sided tetragonal structure consisting of four microsporangia located at the corner, two in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Structures of microsporangium. In a transverse section, a typical microsporangium appears near circular and outline. It's generally surrounded by four wall layers. Figure 1.3b. The epidermis, endothelium, middle layers, and the tapetum. The outer three wall layers perform the function of protection and helping and has of another and to release the pollen. And the innermost wall layer is the tapetum. It nourishes the developing of pollen grains. Cells of the tapetum possess some dense cytoplasma and generally have more than one nucleus. Can you think of how tapetal cells could become binucleate? And the answer is a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporogenesis. As the anther develops, the cells of the sporogenous tissue and undergo meiotic divisions to form microspore tetrads. What would be the pleiady of the cells of the tetrad? As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microscope mother cell. The process of formation of microspores from a pollen mother cell PMC through meiosis is called microsporogenesis. The microspores, as they are formed are arranged in a cluster of four cells. The microspore tetrad, figure 1.3a, as the anthers nature and dehydrate, the microscopes dissociate from each other and develops into pollen grains, figure 1.3b. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. Figure 1.3c. Pollen grain. The pollen grain represent the male gametophytes. If you touch the opened anthers of hibiscus and any other flower, you would find the deposition of yellowish powdery pollen grains of your fingers. Sprinkle sprinkle on these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, sizes, shapes, colors, designs seen on the pollen grains from different species. Figure 1.4. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. If it has a prominent two-layered wall. The hard outer layer called the exine is made up of sporopollenin, which is one of the most resistant organic material known. Now it can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain hexine has prominent apertures called germ pores where sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The hexine exhibits a fascinating array of patterns and designs. Why do you think the design should be hard? What is the function of germ pore? The inner wall of the pollen grain is called the entine. Entine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasma of pollen grain is surrounded by plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. Figure 1.5b. The vegetative cell is bigger, has abundant food reserve and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasma of the vegetative cell. It is spindle shaped with the dense cytoplasma and are shared at this two-celled stage. In the remaining species, and the generative cell divides mitotically to give rise to the two male gametes before pollen grains are shared three-celled stage. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imparted wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. Here, it has become a fashion in our recent years to use pollen tablets as food supplements in Western countries. A large number of pollen products in the form of tablets and syrups it's available in the market. Pollen consumption has been acclaimed to increase the performance of athletes and race horses. Figure 1.6 pollen products. When once they are shed, pollen grains have to land on the stigma before they lose viability. If they have to bring about fertilization, how long do you think the pollen grain retain viability? The period for which pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and some members of Rosacea, Leguminosae and Solanaceae, they maintain viability of months. You may have heard of storing semen sperms of many animals, including humans, for artificial insemination. It is possible to store of pollen grains of a large number of species for years in the liquid nitrogen 196 degrees Celsius. Such stored pollen can be used as pollen banks, similar to seed banks in crop breeding programs. 1.2.2 The pistil, megasporangium, ovule, and embryo sac. The gynoecium represents the female reproductive parts of the flower. The gynoecium may consist of a single pistil, monocarpellary, and may have more than one pistil, multicarpellary. When there are more than one, the pistil may be fused together, syncarpus, figure 1.7b, or maybe free apocarpus, figure 1.7c. Each pistil has three parts, figure 1.7a. Stigma, style, and ovary. The stigma serves as the landing platform for pollen grains. The style is the enclosed slender part beneath the stigma. The basal bulged part of the pistil is ovary. Inside the ovary is the ovarian cavity locule. The placenta is located inside the ovarian cavity. Recall the definition. Recall the definition and types of placentation that you studied in class 11. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in a ovary may be one. Wheat, paddy, mango, and many papaya, watermelon, orchids. The megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule, figure 1.7d. The ovule is a small structure attached to the placenta by means of stalk and called funicle. The body of the ovule fuses with a funicle in the region called hilum. Thus, a hilum represents the function between the ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucleus, except at the tip where a small opening called the micropyle is organized opposite the micropylar and is the chalaza representing on the basal part of the ovule. Enclosed within the integuments is a small mass of cells called the nucellus. Cells of the nucellus have abundant reserve food materials located in the nucellus in the embryo sac or female gametophyte, and ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis, the process of formation of megaspores from the megaspore mother cell, is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell (MMC) in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC, microspore mother cell, undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of four megaspores, figure 1.8a. Female gametophyte in a majority of flowering plants. One of the megaspores is functional while the other three are degenerate. Only the functional megaspore develops into the female gametophyte embryo sac. This method of embryo sac formation from a signal megaspore is termed monosporic development. What will be the ploidy of the cells of the nucleus, MMC, the functional megaspore, and female gametophyte? Let us study about the formation of the embryo sac in detail. Figure 1.8b. The nucleus of the functional megaspore divides mitotically to form two nuclei which move to the opposite poles, forming the two nucleate embryo sac. Two more sequential mitotic nuclear divisions result in the formation of four nuclear and later the eight nuclear stages of the embryo sac. It is the of interest to note that these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not followed immediately by cell wall formation. After the nuclear 8 nucleate stage, cell walls are laid down, leading to the organization of typical female gametophyte or embryo sac. Observe the distribution of cell inside the embryo sac, figure 1.8 BC. Six of the eight nuclei are surrounded by cell walls and organized into cells. The remaining two nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. And there is a characteristic distribution of the cells which within the embryo sac. Three cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergids have special cellular thickness at their micropylar tip called filiform apparatus, which play an important role in guiding on the pollen tubes into the synergid. Three cells are at the chalazal end and are called the antipodals. The large central cell, as mentioned earlier, has two polar nuclei, thus typical and angiosperm embryo sac at maturity through 8 nuclear and 7 celled lie close to each other, so that self-pollination can occur. Some plants such as viola, commonly pansy, oxalis, and commonly produce two types of flowers. Cosmogamous flowers, which are similar to flowers of other species with exposed anther and stigma, and clistogamous flowers which do not open at all. Figure 1.9c. In each flowers, the anthers and stigma lie close to each other. Pollen grains come in contact with the stigma to affect pollination. Thus, clistogamous flowers are invariably autogamous, as there is no chance of cross pollen landing on the stigma. Clistogamous flowers produce a shared seed set, even in the absence of pollinators. Do you think that cleistogamy is advantageous or disadvantageous to the plant? Why? Gynosigamy, a transfer of pollen grains from the anther to the stigma of anther, flower of the same plant. Although gytonogamy is functionally a cross pollination involving a pollinating agent, genetically it is similar to autogamy, since the pollen grains come from the same plant. Xenogamy, transfer of pollen grains from anther to the stigma of different plant. Figure 1.9b. This is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Agents of pollination: plants use two abiotic, the wind and water, and one biotic, animals agents to achieve pollination. Majority of plants use biotic agents for pollination. Only small proportion of plants use abiotic agents. Pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. The to compensate for this under uncertainties and associated loss of pollen grains, the flowers produce enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollinators. Wind pollination also requires that the pollen grains are light and are non-sticky, so that they can be transported in wind currents. They often possess well-exposed stamens, so that the pollen are easily dispersed into wind currents. Figure 1.10. And large, often feathery stigma to easily trap airborne pollen grains. White pollinated flowers often have a single ovule in each ovary and numerous flowers packed into the flower inflorescence. A familiar example is the corn cob, and the tassels you see are nothing but the stigma and style which wave in the world wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. As against this, you would recall that water is a regular mode of transport for the male gametes among the lower power plant groups such as algae, bryophytes, and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, and their distribution is limited because of the need for water for the transport of male gametes and fertilization. Some examples of water pollinated plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine sea grasses such as Zostera. And not all aquatic plants use water for pollination. In a majority of aquatic plants, such as water hyacinth and water lily, the flowers entire emerge above the level of water and are pollinated by insects and wind. As in most of the land plants, in Vallisneria, the female flower reach the surface of water by the long stalk, and the male flowers or pollen grains are released onto the surface of water. They are carried passively by water currents. Figure 1.11. Some of them eventually reach the female flowers and the stigma and another group of water pollinated plants such as sea grasses. Female flowers remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long, mm, are long ribbon like and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from wetting by a mucilaginous covering. Both vine and water pollinated flowers are not very colorful and do not produce nectar. What would be the reason for this? A plant cannot complete their life cycles without each other. The moth deposits its eggs in the locule of the ovary, and the flower in turn gets pollinated by the moth. The larvae of the moth come out of the eggs as the seeds start developing. Why don't you observe flowers of the following plants or any others available to you: cucumber, mango, people, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animal visit them and whether they could be pollinators. You will have to patiently observe the flowers over a few days and at different times of the day. You could also try to see whether there is any correlation in the characteristics of of flower to the animals that visit it. Carefully observe if any of the visitors come in contact with the anthers and the stigma, as only such visitors can bring about pollination. Many insects may consume pollen or the nectar without bringing about pollination. Such floral visitors are referred to as pollen nectar robbers. You may or may not be able to identify the pollinators, but you will surely enjoy your efforts. Outbreeding devices. Majority of flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self pollination results in breeding depression. Flowering plants have developed many devices to discourage self pollination and to encourage cross pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma become receptive and the stigma become receptive much before the release of pollen. In some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self incompatibility, or other flowers of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize monoecious, it prevents autogamy but not gynotomy in several species such as papaya. Male and female flowers are present on different plants, and that is each plant is either male or female dioecious. This condition presents both autogamy and gynotomy. Pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollen. Compatible pollen of the same species as the stigma, often pollen of the wrong type, either from the other species or from the same plant. If it is self incompatible, also land on the stigma, and the pistil has the ability to recognize the pollen, whether it is of the right type of compatible or of the wrong type incompatible. If it is of the right type, the pistil accepts the pollen and promotes post pollination events that leads to fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth or in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of a continuous dialogue. By its acceptance or rejection is the result of continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to precise a pollen tube through one of the jump force figure 1.12 and the contents of the pollen grain move into the pollen tube. Pollen tube grows through the tissues of the stigma and style and reaches the ovary figure 1.12 bc. You would recall that the some plants pollen grains are shared at two-celled conditions, a vegetative cell and generative cell. In such plants, generative cell divides and forms uh, the two male gametes during the growth of pollen tube in the stigma. In plants which shed pollen in the three-celled condition, pollen tubes carry the two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of the ovary, one of the synergids through the filiform apparatus figure 1.12 de. Many recent the synergids through the many recent studies have shown that filiform apparatus present at the micropylar part of the synergids guides and the entry of pollen tube. All these events from pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen pistil interaction. As pointed out earlier, together referred to as pollen pistil interaction as pointed out earlier, followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen pistil interaction even in incompatible pollinations to get desired hybrids. You can easily study pollen germination by dusting some pollen from flowers such as pea, chickpea, crotalina, balsam, and vinca on the glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, now observe the slide under the low power lens of the microscope. You are likely to see pollen tubes coming out of the pollen grains. A breeder is interested in crossing different species and often genera to combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiments, it is important to make sure that only the desired pollen grains are used for pollination and the stigma is protected from contamination from unwanted pollen, and this is achieved by emasculation and bagging techniques. If the female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehiscence using a pair of forceps is necessary, and this step is referred to as emasculation. Emasculated flowers have to be converted with a bag of suitable size, generally made up of butter paper, to prevent contamination of the stigma with wanted pollens. This receptivity mature pollen grains collected from anthers of this male parent and are dusted on the stigma, and the flowers are rebagged, and the fruits followed to develop. If the female parent produces on unisexual flowers, there is no need for emasculation. The female flower buds are bagged before the flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and flower bagged. Double fertilization enter after entering synergies one of the synergies, the pollen tube releases and the two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of the diploid cell and zygote. Other the other male gamete moves towards the two polar nuclei located in the central cell and fuses with them to produce triploid primary endosperm nucleus PEN figure 1.13a. As this involves the fusion of three haploid nuclei, it is termed a triploid fusion. Since two types of fusions, syngamy and triploid fusion, take place in an embryo sac. The phenomena is termed double fertilization, an event unique to flowering plants. The central cell after triple fusion becomes the primary endosperm cell (PEN) and develops into the endosperm, while the zygote develops into the embryo. Post fertilization cell structures and events. Following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds and ovary or into fruit are collectively termed the post-fertilization events. Endosperm development precedes embryo development, while the primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissue are filled with reserve food materials and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the PEN undergoes successive nuclear divisions to give rise to free nuclei. This stage of nuclear divisions to give rise to free nuclei, this stage of endosperm development is called prenuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea, groundnut, pea beans, before seed maturation, or it may persist in the mature seed, example castor and coconut, and be used up during seed germination. Split open now some seeds of castor bees begins groundnut beans, groundnut, fruit of coconut, and look for the endosperm of each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. Embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Through the seeds differ greatly, and the early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Figure 1.13 depicts the stages of embryogeny. In the dicotyledonous embryo, the zygote gives rise to the proembryo and subsequently to the globular heart-shaped and mature embryo. The typical dicotyledonous embryo, figure 1.14a, consists of embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons as is the epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons is hypocotyle, and that terminates at its lower end in the radicle or root tip. The root tip is covered with the root cap. Embryos of monocotyledons, the figure 1.14b, possess only one cotyledon. In the grass family, the cotyledon is called scutellum, and that is situated towards one side, the lateral of the embryonal axis. At its lower end, on the embryonal axis, has the radical and root cap enclosed in an undifferentiated sheath called coleorhiza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyle. Epicotyle has a shoot apex and a few leaf primordia encloses in a hollow foliar structure. And the coleoptile soak a few seeds in water, say of wheat, maize, peas, chickpea, groundnut, overnight, and then split the seeds and observe the various parts of the embryo and the seed. In angiosperms, the seed is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruits. Seed typically consists of seed coat, cotyledon, and an embryo axis. And the cotyledon, figure 1.15a, of the embryo has simple structures, generally thick and swollen, and due to storage of food reserves as in legumes. Mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryo development. Example pea, groundnut, albuminous seeds retain a part of endosperm as it is not completely used. Albuminous seeds can retain a part of endosperm as it is not completely used up during embryo development. Example: wheat, maize, barley, castor, occasionally, and some seeds such as black pepper and beet, a remnant of nucleus nucellus, are also persistent. Now, this residual persistent nucellus is the perisperm integuments of ovules harden as tough protective seed coats. Figure 1.15a. The microbial remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seeds become relatively dry. 1.10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, are germinate. As ovules mature into seeds, ovary develops into the fruit. That is the transformation of ovules into seeds and ovary into fruits proceeds simultaneously. The wall of the ovary develops into the wall of the fruit called pericarp. The fruits may be fleshy, as in guava, orange, mango, etc., or may be dry, as in groundnut and mustard, etc. May fruits have evolved mechanisms for dispersion. Seeds result of the classification of fruits and their dispersion mechanisms that you have studied in an earlier class. Is there any relationship between number of ovules in any ovary and the number of seeds present in a fruit? In most plant, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruits. Figure 1.15b. Most fruits, however, develop only from the ovary and are called true fruits. Also, in most of the species, fruits are the results of fertilization. There are a few species in which fruits develop without fertilization. Such fruits are called the parthenocarpic fruits. Banana is one such example. Parthenocarpy can be induced through the application of growth hormones, and such fruits are seedless. Seeds offer several advantages to angiosperms. Angiosperms, firstly, since reproductive processes such as pollination and fertilization are inadequate independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal of to new habitats and help the species apomixis and polymerase. Then summary. Thank you so much for watching. Please like, share, and subscribe.